Welcome viewers. Today we are going to discuss the process of conjugation in bacterial cell E. coli, where we will see that a plasmid that codes for resistance gene for antibiotics is getting transferred from the donor bacteria to the recipient cell. Before actually starting the process of conjugation, let's have an overview on the gene transfer processes that are undergoing inside any bacterial cell. We all know that gene transfer in bacterial cells do play a vital role, as because this is the pathway wherefrom the mixing of gene takes place within a bacterial cell. As because the bacterial cells are asexually producing, so the only pathway wherefrom they can bring diversity in their genomes is via horizontal gene transfer. Gene transfer in bacterial cell mainly takes place by three important steps. So let's have a look on it. They are bacterial transformation. In bacterial transformation, we can see that the donor cell dies off and it donates its genomic DNA, and that naked DNA gets entry inside the recipient cell. Herein, we will see that there is no direct interaction between the donor bacteria and the recipient cell. And the second step is bacterial transduction. Herein, you can see the involvement of bacteriophages. Bacteriophages are responsible for the lysis of the donor cells, and during their lysis, sometimes instead of packaging their own genomes, they will package the bacterial genomic DNA, and while reinfecting onto a new host or the recipient cell, they will transfer the DNA that they have collected from the donor bacteria and donate it to the recipient cell. And the third one, which is our topic of discussion today, is bacterial conjugation. Herein, we will see that there exists a plasmid inside the donor cell which is absent in the recipient bacteria. And this donor bacteria actually is coding for certain genes which are responsible for its own transfer from the donor cell to the recipient bacteria. But the most important point or most important difference between bacterial conjugation and the rest of the pathways is that cell-to-cell contact is an essential prerequisite during conjugation. So this conjugation event was the discovery of these two eminent scientists called Joshua Lederberg and Edward Tatum in the year 1946. They discovered the phenomena of conjugation while working with two auxotrophic strains, say A- and B-, and when they mixed them together, they got a wild type phenotype.
Ma'am, can you please tell me what are auxotrophic cultures?
Yeah, it's very true. Before getting into the experiments done by Lederberg, we need to define certain things. Number one is an auxotrophic strain. Auxotroph is a strain or a bacterial strain that has got mutation in some of the genes which are responsible for making essential amino acids or essential components which are needed for their survivability. So an auxotroph is a strain that cannot grow in a minimal media. It means the media should be supplemented with that particular component which the auxotroph is unable to synthesize. On the other hand, the counterpart of auxotroph is a prototroph, is a strain of bacteria that can synthesize all the essential nutrients which are needed for its growth, and hence it can grow in the minimal media. So if we look precisely onto the strains that were being used by these two scientists, one strain was methionine minus, biotin minus, threonine plus, leucine plus and thymine plus. It means this is an auxotroph which needs methionine and biotin to be supplied in the minimal media for its growth. On the other hand, the other strain was methionine plus, biotin plus, threonine minus, leucine minus and thymine plus. It means these three things are to be supplied for its growth on the minimal media. So initially what they did, they took 10 to the power of 8 cells from this strain and plated it onto the minimal media. And from this strain also, they took 10 to the power of 8 cells and plated it on the minimal media. But unfortunately, they couldn't get any colonies, as both of them were auxotrophic strains. On the other hand, when they mixed these two strains together, and from that combined extract, they have taken 10 to the power of 8 cells and they were plated on minimal media, they got 10 colonies. So therefrom they could infer that this mixing is doing some sort of event which is converting these auxotrophs into a prototroph. So now there is a question that whether this generation of prototrophs is because of mutation or some sort of genetic transfer has taken in it. So they could defend their results by saying that as they are not getting any colonies from the cultures when they were not mixed, it means the result that we are getting because of mixing is not due to mutation. It is surely due to some sort of genetic transfer being going on between the two cells. Next important discovery that was being done by Bernard Davis in the year 1950 was his famous U-tube experiment. What he did, he took a U-tube wherein it was being separated. There exists a filter in between the two arms of the tube. The pores of the filter were made so that it allows the passage of only the DNA, whereas the passage of bacteria from this arm to the other arm was restricted. And what they did, that in two arms they were having two different auxotrophs, and pressure was created so that the passage of nutrients are allowed, and they took cultures from both the arms separately and tried to plate them on minimal media. But as the previous experiment, they couldn't get any colonies. Therefrom, they could infer that this genetic exchange or conversion of a auxotroph to a prototroph is surely not mediated mainly by transfer of DNA. It means some sort of cell-to-cell contact is an essential prerequisite to carry on the conversion of an auxotroph to a prototroph. So this was supposed to be the two important remarkable discoveries that helped people to understand that conjugation event is something different from transformation. So let's have a look on the molecular events that occur during the conjugation in E. coli. So what happens first, that there exists a plasmid called F factor which codes for a protein called pilus protein. That pilus protein is responsible for bringing the donor cell to the close proximity of the recipient cell. Then it also codes for certain proteins that are responsible for making the conjugation bridge between the donor bacteria and the recipient bacteria. This F plasmid also codes for a class of enzymes called relaxosomes. These relaxosome proteins recognize a specific site onto the F plasmid called ORI-T site, and this ORI-T site is being recognized by this relaxosome, and it creates a nick on the single strand of the F plasmid, and once it creates a nick and separates the strands, then relaxosome contains many proteins. All the proteins are released except the protein called relaxase that remains still bound to this nicked DNA. Next, this relaxase protein will complexed with the DNA. This nucleoprotein complex is being recognized by coupling factors, and these coupling factors are then responsible for introducing this nucleoprotein through this exporter protein. This exporter protein is spanning the inner and the outer membrane of the donor bacteria, and through this exporter protein, actually this nucleoprotein complex, that is the complex between the DNA and the relaxase, gets entry inside the recipient bacteria through the conjugation bridge. Once these things are getting entry, means a single stranded plasmid DNA is getting entry inside the recipient bacteria, then it circularizes taking help of this relaxase protein, and on the other hand the single stranded plasmid that is remaining inside the donor cell also replicates. And the final step is that in both the places, in the donor bacteria and in the recipient bacteria, we do get a replicated copy of the F plasmid. So the net result of F plus cross F minus mating is that both the donor and the recipient bacteria become F plus. It means now the recipient bacteria can also behave as a donor in the next round of conjugation. So these are the basic molecular mechanisms that occur during conjugation. Apart from this, then came the important discovery from Luca Cavalli Forza he could first establish the existence of a newer variety of F plus bacteria. He could find that this strain was capable of producing recombinants at a rate 1000 times more than the rate of recombinants that were found with F plus cross F minus matings. And the second additional difference was that after F plus cross F minus matings, as you have seen just now, that the recipient bacteria is converted to F plus bacteria. So he could find certain strains wherein this crossing event or the conjugation event is not converting the recipient bacteria into a F plus bacteria. Then he started thinking of some other molecular mechanisms that are occurring inside the bacterial cells or inside the donor bacterial cells that are bearing the F plasmid. And the reason or the logic behind this was F plasmid is getting integrated within the bacterial chromosomal DNA, leading to the generation of a strain called HFR, where HFR stands for high frequency of recombinating strain. And as because these strains are being created, whenever this HFR strain tries to participate in the process of conjugation, then it not only transferred the genes that are corresponding to the F plasmid, but genes corresponding to the chromosomal DNA also started to get transferred from the HFR strain to the F minus strain. As a result of which, what we can see here that the recipient bacteria may procure certain genes which are of bacterial host origin, and it gets at some sort of benefit, as here we can see that the recipient bacteria was C minus, B minus and A minus, whereas the donor bacteria where HFR is there is donating its C plus, B plus and A plus strain, and because of this conjugation event, now the recipient cell has become, say for example, B plus and A plus. So the bacteria is becoming more efficient, much more capable of surviving under adverse conditions. So this is the main importance of HFR cross F minus matings.
Ma'am, what is the importance of studying HFR strains?
The HFR strains are very important from the viewpoint of molecular biological studies, as because these strains were being used for the mapping of the genomes. If we just carry on an interrupted mating, then herein you can see that this A plus gene is just being followed up by B plus, then it is just being followed up by C plus. So in this way, if we carry on multiple interrupted mating experiments, then we can get the sequence of the genes which are present inside the bacterial genome. So HFR cross F minus matings are used for the mapping of the E. coli genome. Another fate of HFR is occurring when the HFR tries to excise off its F plasmid. Under such circumstances, we have seen generation of F prime factors, wherein what we can see that the F plasmid is having some genes which are of bacterial origin. On the other hand, some part of the F plasmid is being locked up within the bacterial chromosomal DNA. So these F prime factors are also important, as because this F prime cross F minus matings can provide the recipient bacteria with certain traits of bacterial chromosomal DNA. And here the gene transfer is much faster as compared to HFR cross F minus matings because of its smaller size. The important points in conjugation: that DNA transfer requires cell-to-cell contact, means it is occurring through a conjugation bridge which is resistant to DNase. Next, DNA transfer occurs in one direction, that is from donor to recipient, means the opposite way is not allowed. DNA transfer does not require protein synthesis in the donor cell, means no chromosomal DNA is involved in protein synthesis. Then DNA transfer requires energy in donor cells, primarily ATP. Now let's start the requirements for performing the conjugation experiment in the lab. To start with, first of all, we need to prepare LB broth or Luria Bartani broth. We will dissolve 25 gram of LB in 800 ml of sterile water. After that, we will adjust the pH to 7 taking help of 5 normal NaOH. After adjusting the pH, we will finally make up the volume to 1000 ml and sterilize the media by autoclaving. Standard autoclaving methods. And if we want to make LB agar plates, then onto this media we will be adding 1.5% of agar and carry on the autoclaving. Now we need to use certain antibiotics such as tetracycline and streptomycin. We will dissolve 45 mg of tetracycline in 1.5 ml of 70% alcohol, and we will vortex it to dissolve, and this makes the concentration of 30 mg per ml of stock tetracycline, and the working concentration is 30 microgram per ml. Point is to be noted down that the vials should be covered with aluminum foil as tetracycline is light sensitive. Next, streptomycin. We will dissolve 150 mg of streptomycin in 1.5 ml of sterile water. We will vortex it to dissolve, and stock concentration is 100 mg per ml and the working concentration is 100 microgram per ml. Here we are using two bacterial cultures. One is the donor E. coli strain, naming strain A. It has got a fertility plasmid containing resistance gene for tetracycline. On the other hand, the recipient bacteria is strain B, wherein the resistance gene is already integrated within the chromosomal DNA. So, the instruments and the glassware that are needed while performing conjugation experiment are laminar air flow, as the steps are to be carried on under sterile conditions, incubator shaker at 37 degrees centigrade to carry on the growth of the bacterial cells, spectrophotometer to measure the optical density of the cells, as the optical density is to be measured strictly for successful completion of conjugation experiment, autoclaved sterile culture tubes, autoclaved sterile epigrafts, microtubes, micropipette, sterile petri dishes or disposable petri dishes you may use. Then we have got spreader and ice is needed. First of all, we need to revive the host cells. So, the donor cells should be streaked on LB tetracycline plates and recipient strain B should be streaked in LB streptomycin plates, and the plates should be incubated at 37 degree incubator for overnight incubation. And the next day we will get certain single colonies in both the plates. So, single colonies are to be selected and they are to be inoculated into 6 ml LB broth containing tetracycline for the growth of donor cell and containing streptomycin for the growth of recipient cell. So, these are grown again overnight at 37 degree incubator shaker. After that, from this culture we will take up 1 ml of the overnight donor culture and we will inoculate it into 25 ml of LB broth containing tetracycline, and we will incubate it at 37 degree incubator shaker. Similarly, we will take 3 ml of overnight grown recipient bacteria, means strain B bacteria, and inoculate it in a similar fashion in 25 ml of LB broth containing streptomycin. Both the cultures are incubated at 37 degree incubator shaker and both the cells are grown so that they acquire an OD of 0.8 to 0.9 at an absorbance of 600 nanometers. This is a very critical step. You have to strictly maintain this OD, otherwise you won't get successful results in conjugation experiment. Next, after we have got the desired OD, take 0.2 ml of donor and recipient cultures in a sterile test tube, and both of them are mixed together and that tube is marked as conjugated tube, and we mix it gently and incubate it at 37 degree incubator for 1.5 hours to 2 hours, and 2 other separate tubes as controls were taken, wherein 0.2 ml of only donor bacteria is taken, another tube wherein 0.2 ml of only recipient bacteria are taken. And all these 3 tubes are incubated for 1.5 hours to 2 hours at 37 degrees centigrade. Point is to be noted down that no shaking during conjugation experiment, right? When the incubation is complete, in all the 3 tubes, we will be adding 2 ml of sterile LB broth. And after addition of sterile LB broth, they are again incubated for 1.5 hours to 2 hours. After this incubation is done, that we will take 0.1 ml of cell suspension from each of the 3 tubes, and they will be plated in 3 different types of plates. One plate containing only streptomycin, one plate containing only tetracycline, and a third plate contains streptomycin and tetracycline both. So in all, we need to do 9 platings, means donor strain will be plated in only streptomycin, only tetracycline and containing both, and after spreading the bacterial suspensions, the plates should be inverted and kept inside 37 degree incubator for overnight incubation. The next day, you are supposed to observe the results. So what observations you observe? You will see that the donor strain, which was actually tetracycline resistant, it only gave colonies in tetracycline plates, and as it was sensitive towards streptomycin, it gave no colonies, and obviously in the plate wherein both the antibiotics are there, here also we get no colonies. Secondly, regarding the recipient, we already knew that it contains resistance genes against streptomycin. That's why it gave colonies in LB streptomycin plates. It couldn't give colonies in LB tetracycline plates as because it was sensitive towards tetracycline, and due to this reason only, it couldn't give any colonies in LB streptomycin plus tetracycline plates. Whereas the tube wherein conjugation event has actually taken place, it will give colonies in streptomycin plates as well as tetracycline plates and plate containing all the 2 antibiotics. So getting positive colonies in all these plates is indicative of successful conjugation event occurring, wherein we can see that the tetracycline resistance gene has been transferred from the donor bacteria A to the recipient bacteria B, and this conjugated strain that has now become resistant towards tetracycline and streptomycin both. While performing the experiment of conjugation, certain precautions are to be taken. Number 1, that all the cultures or the media that is to be prepared should be made perfectly sterile. All the steps of conjugation should be carried on strictly inside the laminar airflow, and the optical density of the donor and recipient bacteria should be strictly maintained. And while spreading the conjugated strains, or after completion of the experiment, whatever spreading steps we are doing, it should be done carefully, keeping in mind that the spreader should not be too hot. If it is too hot, then the bacterial cells will die and we may not get any colonies at all. So in today's lecture, if we try to summarize that what we have discussed today is that we started our journey with 3 different modes of gene transfer in bacteria. We have highlighted on the important discoveries that led to the phenomena of conjugation. We have gone through the molecular events that occur during conjugation, and finally we have discussed about the transfer of a plasmid containing resistance gene for tetracycline from a donor strain of E. coli called strain A to a recipient strain of E. coli called strain B that contains a streptomycin resistance gene in its chromosome. And finally, while after performing the experiment, we have seen that the recipient bacteria, which was containing only streptomycin resistance gene, has now acquired tetracycline resistance bearing plasmid by means of conjugation, and it has shown resistance towards finally tetracycline and streptomycin.